According to the World Health Organization, genetic conditions such as cancer, skin conditions, blindness, and deafness affect nearly 60% of the worldwide population. These conditions include sickle cell disease, spinal muscular atrophy, germline mutations, and many more. Most of you have a genetic conditions that you may not know that you have, but can be passed down from you to your child. Some of these conditions ranging from minor to hidden all the way to critical and serious. Genetic editing is being tested and used in today's genetic conditions. Using a technique called CRISPR-Cas9 genetic editing, certain conditions are being treated and cured. Another pro of this technique is that once the DNA is altered, it is passed from parent to offspring, eradicating the condition that could have been passed down from generation to generation. Think about the genetic conditions in your family's DNA that could be removed that are affecting you daily. Genetic editing through CRISPR can be used to treat and prevent genetic conditions that are passed down from generation to generation. This technique is also being tested on non-genetic conditions. As someone who suffers from a genetic condition called Wolff-Parkinson-White, a heart condition which recurs every few generations and will require surgery in the upcoming years, I have been interested in genetic editing since I learned of this technique. Using CRISPR gene editing, this technique is used to modify genomes of living organisms within molecular biology. CRISPR gene editing is a technique derived from the Cas9 protein. As stated by Science Alert, CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. This is a sequence of nucleic acid derived from archaeobacteria that contains copies of genetic bacteria. A protein called Cas9 can be used in conjunction with engineered CRISPR sequences to hunt down codes and slice them like a molecular scalpel, allowing geneticists to cut out a target gene either to remove it or replace it within a new sequence, announced from the article Science Alert, which is CRISPR gene editing. When the Cas9 protein finds the virus's DNA strand, it would break the DNA strand and save a certain sequence to remember the virus for future instances. This protein was discovered by scientists and provided insight on a future where genetic editing could be possible. Scientists are using the Cas9's protein's ability to cut and remember viral DNA. They then engineer a sequence of DNA to take place of the genetic condition within a living organism's DNA. Scientists use Cas9's ability to hunt down viral DNA and cut it out of a DNA sequence. Identifying a certain condition's DNA sequence, scientists give a copy of the condition to the Cas9 protein and insert it within the host of the genetic condition. Upon removing the DNA sequence containing the condition, scientists then insert a new healthy template into the host DNA sequence. As we know, DNA replicates its original 2 sequence into 2 DNA strands. Being that the original DNA strand was altered, all other DNA strands following the replication process will contain new DNA sequence, diminishing the genetic condition within the genome. This technique is being used today through a number of crippling genetic conditions. In the Netflix documentary Human Nature, a young boy, David Sanchez, suffers from sickle cell anemia and is placed into a CRISPR gene editing trial. In the 2018 article, Target Delete Repair, CRISPR is a revolutionary gene editing tool, but it's not without risk. Author Mark Schwartz states, with sickle cell anemia, symptoms such as periodic pain, infections, weakness, delayed growth, and vision problems do occur. He also states, sickle cell anemia patients within a 70% threshold of healthy cells live symptom-free. The treatment, which is being used as a blood transfusion once a month, kept David at the threshold and he now lives symptom-free. Being that this is still a new technique, there are still many risks and testing required to make this a safer and reliable technique. A recurring issue with gene editing is the deletion of a targeted DNA sequence. When selecting a targeted DNA sequence, scientists have to be precise and sure when deleting a specific DNA sequence. Accidental deletion of a non-targeted DNA sequence can result in mutations, infections, and diseases. For example, in the 2020 article by Associated Press found on Stats News, researchers made 40 embryos with eggs from healthy donors and sperm from a man with a gene mutation, a single letter missing in the DNA alphabet that causes blindness. Correct deletion of a specific sequence could even cause issues by removing bacteria that is vital to a genome's immune system. Genetic editing is still being tested on stages of the human genome. Scientists have been testing this technique on embryos, younger children, and adults to learn the risks that could progress over time. Embryos are safer to test on because the human is not fully developed, but reports show that remaining uncertainty about the prevalence, extent, and detection of genetic damage and mosaicism still cause risks. As stated in the 29 article by Benjamin Daves, The Technical Risk of Human Gene Editing, mosaicism is a condition that affects any cell in the body. Testing this technique on children, young adults, and adults proven to be risky and cause cancer by the accidental deletion of a DNA sequence. Testing on elderly has proven to be useless because their bodies are developed and the aging process and the degrading of the cells do not allow this technique to be processed throughout the host. Being that this technique is still very risky and should only be practiced by medical professionals. In recent years, the speculation of changing the human genome and making our species a more perfect species has been tested. This area of trials can be risky when relating to changing our species, completely eradicating flaws, conditions, and making us all similar. Genetic editing doesn't just stop at genetic conditions, it could also bring cures and treatments to today's common illnesses and help shape the genome of not just humans, but all living organisms. Using the CRISPR gene editing technique, trials have begun to use this technique to treat common illnesses, diseases, and critical medical cases. Using the Cas9 protein's basic ability to help fight common infections, this technique is being implemented by giving the protein segments of today's viral DNA sequences and program the protein to remember and fight these common illnesses. Being that the cancer is a critical genetic issue, CRISPR can be used to remove this DNA sequence from the human genome if not genetic. This technique can still be implemented to a host to progress the human immune system. Genetic editing is still being tested on embryos, animals, and even plants to change our way of life and make the living organisms world more perfect. This technique is being used on animals that we use in today's human transplants to engineer their organs to be more acceptable to the human body, lowering the risk of rejection. Fertilized embryos' DNA is being altered to change the results of the fully developed fetus before and after birth. This supports the controversy of designer babies. Along with altering the DNA of embryos, it is said that potential intelligence, body figure, and emotions and personality are all within DNA segments. In conclusion, genetic editing can be used to treat and prevent genetic conditions that are passed down through generations. This technique is also being tested on non-genetic conditions. Bottom line is, genetic conditioning does have its risks, just like every medical procedure, techniques, treatment, and many more. Personally, I believe this was a God-given right to do good for our society and help change the future and make it safer, healthier, and proficient future of health. Genetic editing is the future of curing genetic and non-genetic conditions and shaping the future of healthcare. This technique will save countless lives and be carried on through generations. Thank you.